The overall goal of this protocol is to quantify the proportions and absolute levels of STAT3 splice variant transcripts in eosinophils.
This method can address key questions in the splicing regulation field, such as co-association of distal splicing events and identification of conditions that can cause tandem splicing proportions to differ. The main advantage of this technique is the plasmid standards enabling acquisition of absolute and relative qPCR data, so we can determine proportions and overall levels of 4 STAT3 transcripts.
In our case, we wish to learn how the 4 STAT3 variants, S-alpha, S-beta, delta S-alpha, and delta S-beta change when eosinophils are stimulated by cytokines.
Though this method can provide insight into eosinophil STAT3 composition, it can also be applied to other cell types for which cDNA can be obtained.
Because S and delta S vary in only 3 nucleotides, we had to sacrifice efficiency to achieve specificity and needed standards for each variant.
In this protocol, cDNA prepared from human eosinophils is used as template for amplifying the STAT3 splice variants. After creating the plasmids as described in the text protocol, prepare sequencing reactions for sequencing plasmids from several colonies. Each 20 microliter reaction should contain 3 microliters of sequencing reaction buffer, 2 microliters of sequencing reaction mix, 12 microliters of ultrapure water, 1 microliter of plasmid, and 2 microliters of sequencing primer. When the sequencing results are available,